Good afternoon, everyone, and welcome back after the break. So in the first part of the course, we heard about various ways to calculate models and find structures and models in the rcsb.org or from other data resources. What we're going to focus on in this session is visualizing that structure or structures that you have identified or computed. Now, this is one of the most fundamental things that you probably want to do with the structure is to see overall what the shape of the molecule is, and then also examine closely specific regions of that particular structure, perhaps a particular residue, how a ligand is binding, where the interaction faces between your protein of interest and its partner protein or nucleic acid. You want to also examine and understand how mutations may impact the structure and interactions of a protein. So all this, to do that in a visual way, the tool that we're going to use here is MolStar. And I'm going to present MolStar as a tool to examine one particular structure. That's where we will begin. But then I will also show you a standalone MolStar, and the relevance or importance of that will become hopefully evident when I get to that point. And then finally, I'll also show how the MolStar tool has been woven into various tools and functionalities that the rcsb.org website offers, so that you can not only look at the structure, but also map on it various annotations. You can look at trends and patterns and things like that, that might give you insights into the function and biology of your protein or molecules of interest. So these are the three things that I plan to cover in the session here. And let's get started with the first part, where I'm going to talk about exploring one particular 3D structure. And in this case, I'm going to start from the structure summary page that was introduced to you in the previous sessions. And to do that, I'm going to go back and forth between the slide deck here and the website, actually, so that you can, if you are in front of a browser where you can do this, you're welcome to follow along. So this is the rcsb.org homepage, and let's just start with a simple example. 4-HHP, this is hemoglobin. This is not an Arabidopsis or M. luteus or any other such protein. We'll come back to an Arabidopsis example later, but this is a quick way for me to introduce you to the tool. So how do you access this MolStar tool? So on any structure summary page, whether it is an experimental structure or a computed structure model, the 3D view tab, this will open up the structure, or at the bottom of the page, at the bottom of this little graphic, there is this link called structure that will also take you to that same page. And I wanted to spend a couple of minutes getting you oriented to this. So this is the MolStar interface, and the white space here where you see this colorful molecule, you can use your mouse to rotate things, whatever is being displayed here. This is our 3D canvas. On the top up here is our sequence panel. So notice for every polymer that is displayed in this, the sequence of that is shown up here. And if you click on something there, the same is identified and clicked here. So if I click on something here, notice in the sequence panel, it gets highlighted. So there is a connection between these two. This panel on the right is the controls panel. The controls panel allows you to do many different things. The top part is telling you something about the structure. So are you looking at the assembly? This is the default, but you can also look at the symmetry mates. Since this is a crystal structure, it computes the symmetry mates. You can go back to the assembly. The measurements and other panels we will come to in a little bit. The structure motif panel was already demonstrated by Brinda when she showed how you can select specific residues and do this. But let's start here with this components. The components is actually showing you what is being displayed here. So there are polymers. If I hover over this, it is highlighting all the polymers. I can hide that or show that. There are ligands. So in this case, this is a hemoglobin molecule. So the heme ligands are here. So I can hide that and show that. Waters, the same thing. and an ion. So there is I think, a phosphate ion in this structure. So that's a separate component. Now, this is the default display. I can create any combination of ligand and residue or different chains and create components out of them. And I'll show that in a moment. But those can be added to this list and they can be independently turned on and hidden and things like that. If the particular structure has any experimental data, such as electron density or EM map, you can turn that on here. The quality assessment, if you remember, Brinda talked about the PLDDT scores. So PLDDT for computed structure models and the EM sorry, not for EM, for X-ray structures. There is a quality assessment that is available for display here, which not in this, but I think I can show it to you in 2D and 2. That's another structure. This is another hemoglobin structure. And in here, if I look at the quality assessment, and so the blue regions are the higher quality and the orange and yellow regions are the lower or less ordered regions. And this is being designed to mimic or be similar to that PLDDT coloring scheme that you had seen earlier. So let's go back to our 4-HHB. And okay, further down here, you have options to export animations. You can export the model and do a bunch of other things. So all these functionalities are covered in the controls panel. And again, this is your 3D canvas and sequence panel. So back to our, so these were some of the key features that I wanted to point out. We've already covered all of them. So let's go on further. Now, when you upload a structure into your tool, perhaps the first thing you want to see is what does this molecule look like overall? Now, the molecule may be a single protein chain or a complex of multiple protein chains with nucleic acids and carbohydrates and whatnot. So you want to see the whole thing first and see what all is going on in here, and then perhaps zoom in and try to understand what is unique to what is happening around a ligand or a particular active site or whatnot. And so the MolStar tool functions in two different modes. And so let's go to our demo session. So here, if I, in the default mode, if I move this around and click on, so I'm clicking on this heme, you can see it's showing up as heme 142 at the bottom. If I click on this, it'll jump in, center around that particular heme, and show all the amino acid side chains within 5 angstroms of that particular ligand. It'll also show you through these dashed lines the types of interactions that are stabilizing or holding that ligand in place, right? And so this is very easy and a quick way to understand what is going on in the neighborhood of your ligand of interest or a particular residue. I can go to any residue and say, I want to see what is going on in the neighborhood of this residue, okay? So you can go to any residue. So this is the default mode. But so this is great if you want to pick a particular residue and say, okay, show me what's going on around here. But sometimes you may want to select a number of residues. So what do you do then? You can't keep jumping around. And so for that, you activate the selection panel by clicking on this toggle menu, this little arrow. Notice here, a new menu has now shown up. And with this new menu turned on, now I can either click on a number of residues in here and see those are getting selected in here, or I can click on anything on the 3D panel. And if I click on a white space, all the selection is gone. So this is a quick way for selecting things. And then I can say, I want to do something with this. So why would you want to select? You've selected and you may say that I want to color it in a different color, or I want to see it in a different representation. So for example, let's look at this particular heme. So I'm going to select this heme. And I want to display this in a space fill representation because I want to see how much volume does it really occupy in this cavity that that it is sitting in. So to do that, I can click on this and say, change the current selections representation to space fill. And I'm going to give it a name. So I'm going to call it heme1 and create component. So voila, now my new component, notice a new component was introduced here, which is showing this heme group in a space fill representation. The components do not have to be a ligand. They do not have to be a polymer chain. That can be any combination of chains, residues, whatever it is that you want to look at in one go. And I can hide this and show this. And depending on what it is I want to do with this, so perhaps I'm trying to create a publication quality figure, I may want to change the color of this and do all kinds of other things. To change colors, I can click on this and say, I want to color the whole thing yellow. Okay, things like that. And I can hide that. Okay, so we learned how to quickly look at the neighborhood of a particular selection. And also we learned how to select different things. And so these two, focus and select, these are two things that we are able to do. And we learned about that. Now let's go to the next thing. We also actually quickly looked at how to color things, how to create new components, and how to select things. So the selection, if we go back to our thing, I can say that in this, I want to select everything that is a ligand. So it's asking me which ones do I want to select, and I can say I want to select in one go, I want to select all the heme groups. So it's picked up all that. Okay. And similarly, there are other ways of selection that you can use using the sequence panel, where I can go in and say, okay, I want to select all the hemes. And so now I unselected it. So there are several ways to select things and several things that you can do once you have selected your residue or chain of interest. The selection level, as you may notice, right now we were in the residue level. We can select chains, entities, or go, more granular. So we can even select at the atom level, and we'll look at that in a moment. Okay, so we've learned how to display color. Measurements. Now, one other thing that you may want to do when you're visualizing your structure, you may ask questions like, how far away is this atom from that atom, or this ligand from that ligand, or this chain from that chain, and so on. So for example, let's go back to the beginning and say, so we know in the case of hemoglobin, there is this proximal and distal histidine, which is sort of holding that heme group in space. And I wanted to find, or let's ask this question, how close is this heme group to this proximal histidine? And so the easier way to do this would be I can just jump in here, click on this, jump in here, and zoom in to focus in around the heme. Now, the distance that I want to measure is this distance. Okay, so I'm going to activate the selection mode. And instead of being at the residue level, I'll go to the atom level. So now when I click on this atom and that atom, I have selected just the atoms, not the whole residue. And in the controls panel, if you open up the measurement, you will see that these two selected atoms are already showing up. In order to label, I need to select only one atom. That's what's listed here. For a distance, I need two atoms, and I've already selected the two atoms. And if I click on distance, it is showing up as 2.14 and 2.14 over here. And if I wanted to do more than that, if I wanted to measure an angle, I would have to select 3 atoms, and for dihedrals, 4 atoms. And so this is another quick way of doing some analysis to see what your structure is telling you about the function or the biology of that protein. It is possible that under different conditions, in the presence of different ligands, cofactors, environmental conditions, etc., that distance may change. Those bonds may become, longer, shorter, whatever. So here's a quick tool you can use to measure and compare things. Okay, so now what I'm going to do is all the things that I was showing so far, they were directly interacting with the structure. So you open the structure in this 3D canvas, and you were trying to examine the neighborhood, measure distances, change representation, so on and so forth. But can I use this tool to learn more about what is known in the scientific literature or from other data resources about this particular protein or assembly? And so to do that, let's go back to the structure summary page. And notice this 1D, 3D view. This will actually, if I click on this, this will open a view where on one side, the sequence and 1D annotations are provided. On the other side, there is this 3D structure. And these two are connected so that if I rotate this and click on something here, it gets highlighted here. So what can I do with this? This top part is information derived from the structure. Things like, outliers and binding sites. So where are the heme ligands binding? Where is this? So right now I'm looking at chain A. Where along the chain A are the chains B, C, D interacting? So those are highlighted here. But in addition to that, information from structures, if I scroll down, I can actually see things, annotations that are being brought in from PFAM, CATH, and various other resources. So for example, here is a glycosylation site. And again, I can zoom in to look at the residues. And so this is a lysine that is glycosylated. So if I click on this, in the 3D space, it'll actually jump into that lysine and show me. So this particular structure does not have the glycosylation, but I can quickly examine that where is this residue located in the overall structure, and what would happen if this was glycosylated. So here, right now, there is some other residue and thing, and so I don't want to get into the details, but I can turn this off. And using this left-hand panel where the various annotations are, I can look at what is going on or where are those particular residues or interactions located in the structure. Okay, so this is helping us gather information from other resources onto the structure, map it, and then maybe that'll help us ask new questions or develop hypotheses, etc. So again, as a quick summary, on the left-hand side, there was the sequence panel, the 1D panel, this is the 3D panel, and they are connected in a manner that if you click on either, you can select residues on both. On the top part, we saw structure features. On the bottom part, we saw various annotations from the various data resources that we connect with and integrate information from. So this is all that I wanted to talk about in terms of the examining one particular structure, right? And although the ligands, et cetera, will not be available for CSM, the computed structure models, because they don't have them right now, but other annotations and other things that you can do, the same functionality of 1D, 3D connectivity is available for those structures too. Now, the next thing that I want to point out or show is a standalone MoleStar tool. Now, if you haven't already figured out, what I started with was from the structure summary page, but what if there is your model is not in the PDB, or it's not an experimental model that has been submitted or a computed model that has been submitted to a database? You can still, so if you go to the home page and under visualize, click on MoleStar, it'll actually launch a standalone MoleStar tool for you. So the MoleStar tool here is not connected to any structure summary page. You could upload or import files. So you can open a file from your computer, you can upload a file, or you can even call on a PDB entry. So here, for example, I'm going to call on 4-HHB again and click on apply. So that same thing came up. But what I'm going to do on top of this is that I'm going to also call the AlphaFold structure. And I happen to know that the UniProt ID for the alpha chain of hemoglobin is P69905. I'm going to call that. And this lighter green structure that has shown up is the alpha fold human alpha chain, hemoglobin alpha chain, but they're not quite aligned, and I don't know what to do with this now. So, but the tool allows you to do an alignment right here. So what I can do is I can select the two alpha chains. Now, instead of being at the residue or atom level as we were doing for measurements, I'm going to go to the chain level. So if I click on any part of this lighter green, the whole chain gets selected, and the darker green, the whole chain gets selected. So I have two chains, two alpha chains, hemoglobin alpha chains that are selected. I can now say structure superposition by chains. These are the two chains that are selected, superpose, and voila, it jumps and it is superposed. At the bottom of this 3D canvas, the RMSD is reported. It's very close. But one thing I wanted to point out is that look at the N terminus here. Sorry, I have to get out of the selection mode. I'm going to click on this. Why is it not showing up? What I wanted to point out to you is that notice the alpha-fold protein starts with the uniprot residue 1, which is a methionine, but the structure, the experimental structure actually starts with the next residue, which is a valine, I believe. I don't know why this is not working. Anyway, and so this idea actually came up in one of the questions that someone had posted in the Google Doc, is that when we are looking at these models, they are actually modeling pretty much the whole sequence, including any signal peptides or residues that are removed after the protein has been expressed that may or may not coincide with what the expressed protein's experimental structure shows you. And so we should be mindful of that and be aware and pay attention to these little details, because that may have implications in where the functional sites are and how the ligands interact, etc. So I showed you how to upload coordinates. I showed you how to compare structures, and this is what we did. This is the exercise we did so far. And I'm going to shift gears to now looking at not just one or two, but large collections of structures. And to do that, I'm going to go back to our structure summary page. In the structure summary page, so I'm back in the hemoglobin structure summary page, and here what I wanted to point out is that we did a search where we searched for all structures in the PDB that have a particular uniprot ID. But what I'm going to do here now is I'm going to pull up all those instances or all those structures that have that specific uniprot ID as not a list of structures, but as a group, as a collection. And what that entails, so I can access that either here or over here. So this is a uniprot group or uniprot group here. I'm going to click on this. And so it's telling me that there are 311 such structures where this particular uniprot appears. It tells you when these structures were released over time. It tells you whether they are all, the majority of them are X-ray structures, some electron microscopy, neutron diffraction, one computational model structure also, and it tells you other things. What is interesting is if I click on this group sequence tab, so this is like our 1D view where it is showing the sequence of all the chains, all the polymer chains that correspond to this particular uniprot ID in all these different structures. And notice it's not just one instance, all the instances are listed here. So this _1, _2 is telling you that. Some annotation gathered from other data resources are mapped here. You can click on structural features like cath and SCOP, etc., telling you where the different domains are, which parts are helical, which parts are not. Binding site, this tells you some interesting things about where all these different ligands interact. So some of these ligands interact with this region, some of these ligands interact with this region. And of course, we are showing a linear presentation, but actually this is a 3D structure. And so to truly appreciate this, what we can do is actually open that 1D, 3D alignment. And so if I click on this, so this is similar to that 1D, 3D view that we had seen earlier, but now we are not looking at one structure, we are looking at all these structures. So everything that has the uniprot ID that is appearing in any structure is listed here. The top structure, the top row shows you the AlphaFold structure. So this is already, displayed for you, but I can turn on any number of structures. So this is again showing you just the matched region, along with the whole of whatever else is in the structure, along with ligands. Okay, so I think that this gives you a very easy way to turn on, and you can turn on as many structures as you want, and look at all their ligands and all their structural, whatever other chains that are present in here. So what this also offers you is, if you scroll down, notice here there are these little pink blobs. These pink blobs are indicating mutations. So if I click on this and zoom in here, so I'm going to hide some of these for clarity. So the AlphaFold structure has, if you can see at the bottom, so as I mouse over, the AlphaFold structure so this is valine and this is glutamate. So there is a mutation, and this is what the result of the mutation would be. So all these structures are superimposed. And I can scroll down in this list and see that there are other instances where there are mutations here, and what those mutations are, how they compare to this other mutation that I was displaying. All this is possible just on one screen by turning on and off these different buttons. And I think this is a very powerful way to look at trends and patterns and see how things are changing in the face of whatever structures you're looking at. So with this introduction, I'm actually going to shift gears. So this is what I showed you. I'm going to shift gears towards this Arabidopsis thaliana resistosome story. So we already saw in our earlier before break talk how we can find structures of this Arabidopsis resistosome. And we came upon this structure earlier. But I wanted to actually start here and ask some of these questions. So this looks like a pretty complicated assembly. Which one of these is Arabidopsis is this resistosome protein? What does this protein interact with? How can I figure that out? What ligands does it interact with and where? And can I learn something about mutations? How do I do other analysis? So I'm going to quickly go through this. And I know I'm running out of time, I hope. So the PDB ID is 6J5T. 6J5T. So to quickly scroll through the page and look here, I can see that the third entity with these chain IDs is the resistosome protein. And if I look at, so you can look at the 1D, 3D and look at the whole structure, but I actually wanted to open, or let's do that quickly, just so that. So this is the structure that I showed on the slide, but I can quickly click on this to see where the interactions are with this particular ligand. This is where the ligand is bound. I can look at various mutagenesis points and notice where the ligand binds, there are mutagenesis points. So clearly mutations in those residues would interfere with the protein's function. And as I mouse over this, notice on the top right, it tells you how there is increased sensitivity over here, if you notice. There is increased sensitivity to such and such tissues if there is a mutation. So things like that. So you can gather all this information. What I can also do is for that, if I pull up that whole group thing and look at that 1D, 3D alignment. So this is again the AlphaFold structure for that protein, but I can open up. This particular structure looks very similar to the AlphaFold protein. This part is missing, that's a different matter. But if I look at this structure, it's not as well aligned. And so that's surprising because it's the same structure, same sequence, same protein. And if you remember from the overview slides that Dennis had presented, there is some conformational change going on. And in order to accommodate the conformation, so this particular algorithm for superposition does not give you a good sense of that. But what I can do is I can go to pairwise alignment and I can say, I want to look at 6J5T, and I'm going to look at this AlphaFold. Sorry. I'm going to just copy it from the document because I believe I have put it in there. This one. So let's go back to this. And instead of doing the default, I actually want to do the FatCat flexible comparison. And so the default was not working as well as we wanted it, but now this actually does work much better. And oh, I aligned the wrong chains, but you get the idea that I'm trying to say that, you know, you can use a tool like this to inspect and analyze which chains are relevant to your topic of research. And then you can use the other tools that we offer, like this pairwise alignment, using the different options that are available to get a better match or a better understanding of what is going on. So that is pretty much what I wanted to cover. In the notes, I've listed all the tools that I had used. So we looked at the single structure analysis, we looked at a standalone MoleStar, and we looked at how viewing groups of structures and their 1D, 3D alignment can give you information.